Okay, I'm alive. Good morning to you guys. I know you have started your third lecture test. Looks like the voice is not in sync with the lips. Don't know how to correct that, but hopefully you can hear. Anyway, we're going to start chapter 10 on page 339, and this will be on your fourth test. We'll have 10, and I believe it's 11 for the fourth test. So I've got a Word document done for you. I'm going to mail that pretty soon. Going to go over it a little bit more and make sure everything's pretty much cleaned up. But as you look on page 330, you see muscle tissue and physiology. So we're going to learn the anatomy of a muscle, and then we're going to learn the physiology. You'll find this to be, I think, very interesting, but challenging. So anyway, page 339, if you look down at the bottom there where it says overview of muscle tissue, and you come down to the paragraph, you come down to the second paragraph, right after the title overview of muscle tissue, and you see the bold print term muscle cells. So they can be called myocytes. They can, of course, site being the word for cell, or they can be called muscle fibers. They can be called muscle cells. So you got three names there that mean the same thing. So let's go over to page 340. And you look in that first column, and it mentions what you already know to some degree. You know the three types of muscle. You know, they call them skeletal, cardiac, and smooth. Remember, smooth is not a location. Skeletal tells you it's a location. Cardiac tells you it's a location. Smooth doesn't tell you that. Visceral is the word I'd like for you to use. Visceral is the term for your organs inside. So when you talk about visceral muscle, you're talking about muscle that is part of the composition of, say, your GI tract. It's got connective tissue in it and glandular tissue and so forth. But anyway, we're going to call it visceral muscle. Smooth is a word that describes the appearance of the muscle. Now, as you look at skeletal muscle, which is what we're going to spend most of the time on, we'll mention a little bit about visceral muscle toward the end. But as you look under types of muscles, you come down under skeletal muscle and cardiac muscle and smooth. They call it smooth and so forth. And the next sentence says, a skeletal muscle is made up of long, multinucleated muscle cells that are in parallel, like your fingers are parallel to each other. So you look down at the bottom of the page, and you've got figure 10.1, and you see the illustrations, colorful illustrations. And the skeletal muscle is on the left. You see it looks like tubes, but notice it's got several nuclei presented in the cells. Each tube that you see, each tube-like structure is a muscle cell. So they're multinucleated, and you see the little stripes in there. So we call them striated muscles in terms of description of the appearance. They are striated or have stripes on them in a sense. You go to the middle illustration and you see cardiac muscle. And the first thing you notice, of course, is it's already striated too. So they're similar in that respect. But notice that the cardiac muscle fibers branch. Skeletal muscles don't do that. So they branch, and you see they basically have one nucleus per cell. And then you see this little structure called an intercalated disc. And that is a structure that joins cardiac muscle cells end to end, and it actually helps helps the tissue to perform as one. So it's as you look back up in the paragraph right above it, you'll see it's called a gap junction, but it helps that cardiac tissue to work as one muscle. Okay. And then you look to the right, you see it's got down at the bottom smooth muscle. We would call it visceral muscle, found in things like your GI tract, found in, found in your bladder, it's found in your ureters and so forth. When you start to swallow, you got some visceral muscle here in the esophagus, and so pushes that food down to your stomach. That's how food gets down there. Sometimes people have a hard time getting that muscle to work like that. But anyway, that's a visceral muscle, and you see there are no striations. So visceral muscle does not have the striated pattern that cardiac and skeletal muscle have. I'm going to let you guys read the properties of muscle cells: contractility, excitability. It can be excited, can conduct the action potential. We talked a little bit about that back in chapter 3, I think it was, when we talked about a charge on either side of the plasma membrane. Positive a little bit on the outside, a little bit negative on the inside. So there's a gradient there because of the ions. We want to keep that in mind because that's going to come into play in terms of getting a muscle to contract. And then you see extensibility on page 341 and page elasticity. Nothing hard about that, just you want to be familiar with them. Now we come to structure of the muscle cells. And so I think I look down here. I'm looking at the big thing there instead of the little thing down here on the surface. So you see structure of muscle cells. If you look at the bottom of page 341, you see there's a comparison between what we would call a generic cell. Could be a cell of your GI tract, like a columnar epithelial cell. That could be a cell out of the tubules in your kidney. That would be a cuboidal cell. And this one kind of looks like a cube, doesn't it? And you see it's got the structures that compose that cell labeled. And then you look over to the right and you see what a muscle cell looks like. Quite a bit different in terms of its shape. And you see all these structures inside there. But before we get to those structures, as you look at the comparison of the two, you see plasma membrane. Look over to the right, you see sarcolemma. That's the cell membrane of a muscle. It's a phospholipid bilayer. Got proteins floating around in the membrane, just like we saw back in chapter 3. Then you come down to cytoplasm. Remember how some people call it cytoplasm, some people call it cytosol. In the muscle, they'll call it sarcoplasm. That's okay. You may hear them slip in a cytosol or something like that. It's all the same thing. A couple of different words for those pieces of material. Then you come on down to smooth endoplasmic reticulum, and you look over to the right, and there's sarcoplasmic reticulum. Sarco is associated with flesh, okay? So that's the blue tubes that you see on these little round structures. And then you come down and see mitochondria. Mitochondria are present in both of them. What does mitochondria do? What do mitochondria do? That would be the proper English there. Then you see a nucleus. So what you see is very similar in structure, except you see one different thing, and it's the myofibril. Now, the muscle fiber is a cell, but inside you find these tube-like structures that are called myofibrils. We'll get into that pretty heavy, probably heavier than you want to consider. But anyway, myofibrils are the organelles that are found in a muscle cell, and they have their functions. So as you see on page 341, the second column, look at the middle of that, come down to where it says quick check. Think about how you can differentiate or talk about how similar liver cells are to muscle cells. Now, let's look on page, I think I'm going to get you to go over to, let's do this. Let's go over to page 345. I think we'll be okay there. Page 345. And you've got this big picture, this figure 10:6. That's what it says, big picture up at the top. And you're expected to know the structures that compose a muscle. So as you look to the left, you see you got a bone, and then you see you got this white tissue called a tendon. Now, remember how I've asked you to go back and review Old Testament material? Well, here's a great question. See, here you have a tendon. What's its purpose? And of what kind of connective tissue is it composed? You want to be able to tell me about that, see? So you see there's a tendon, and then when you get into the reddish part, that's the muscle. And if you notice, there is a sheath over the muscle, kind of a grayish area. You can see where it ends a little bit toward the right, and you see epimysium. And of course, epi means on top. And so what kind of connective tissue is that? So that's part of that covering of the muscle. And then as you look at the cut area where they cut the muscle, let's say in half, you look inside the muscle and you see these sections, don't you? Some people would call them bundles. That's okay. You can call them that because there's a bunch of stuff in there. And you see this gray stuff between those bundles. Now, come to not to the bottom of the page. We're still on this top image. But come to the bottom of that image and you look to the right, and you see this term called perimysium. Peri, of course, means, well, you tell me what it means. But you see how it surrounds some sort of bundle of things. You can't really tell from our view here, but that's another type of connective tissue. As a matter of fact, it's the same connective tissue that the epimysium is. It's just inside, and it separates these bundles of structures. Now, one of these bundles is protruding, and you can see it's got a sort of a grayish color to the outside, and you look down below there, you see there's another arrow pointing to perimysium. So these bundles in here are called fascicles. And so the fascicles contain, if you look at the cut end of the fascicle, you can see what's called a extending past that cut area, and that's called a muscle fiber. And if you look closely at the cut face, not of the muscle fiber, but look at the fascicle, you still see gray material dividing some things up. Well, it's actually dividing up and collecting, holding what we would call muscle fibers or muscle cells. But that tissue in there that separates those muscle fibers is called an endomuseum. Same connective tissue as the epimysium and the perimysium. So that type of connective tissue, I'm not going to tell you what kind it is, I almost said it, but that type of tissue, they're all the same. A particular kind of connective tissue, very strong, pulling, okay, very strong. As you know, people can lift some very heavy weights, and that puts a lot of stress on the bones and on these connective tissues as well as on the muscle tissue itself. So as you look at the cut face of the fascicle, you see 1, 2, 3, 4, 5, 6, 7 bundles in there. 7 bundles of some structure. We'll get to it a little bit. But those are actually muscle cells, myocytes, muscle fibers. And you see that extended muscle fiber. So what we want to do is we want to follow this arrow. As you come out to and follow that extended muscle fiber. You can label this as you go along, call it a muscle cell, myocyte, or whatever. Follow that arrow down, and now you see the word muscle fiber. So we're looking at a muscle cell in the second image on page 345. Now, you come to the left of that second image and you see the word Endo Museum. So we have epimysium, perimysium, endomysium. Endomysium surrounds the individual muscle fibers or muscle cells. Now, as you look at this cutaway view of it, you see the blue material in there. We'll talk about that in a little bit. You see the yellow in there. We'll talk about that. And then you see this little protrusion or this extension, I could say. And you can even see the striations in there, can't you? The alternating bands. All right, so now we have a muscle fiber here that's got some cutaway material. And the piece that's sticking out is what they call a myofibril. A myofibril. That's one of those organelles inside a cell, muscle cell. They float in, you can call it cytoplasm, you can call it sarcoplasm, you can call it cytosol. It's all the same stuff, just different names. So here you have this extension, and then when you look at that extension, you can count those and see how many basically are in a muscle cell. I'm going to count the long one first. I see one. I'm moving to my left. 2, 3, 4. You can see they're cut. They're flat cut off. 1, 2, 3, 4. Then you move back to the right, and you see 5, 6, 7, 8, 9. Let's see, 5, 6, 7, 8, 9, 10. I think you got about 11 or 12 in there. 11 or 12 myofibrils in each muscle fiber or in each myocyte. Very important organelle. And we'll get to the blue material and the yellow material in just a moment, but we're going to deal only with the myofibril. So as you come down where that arrow drops again, and now you see it identifies it as a myofibril, and that's one of the organelles. Let's say there's one of the 11 or 12 organelles inside a muscle cell. Now, they give you a section of the myofibril. As you look at that section, you see sort of a pink line to the left, and you look to the right, and there's the same kind of a light pink. Now, you girls have a better idea what color is. My wife is great on color. She said, let's paint the Den white or whatever, and I go in and pull a can of white down. She says, no, that's Jacobean almond. Okay. My observation of color is not nearly as good as hers. So anyway, whatever color you want to call it, that's all right. But you can see there's a light area, a light band to the left, and there's a light band to the right. And then you see in the middle a darker band. Got some little lines in it, and we're going to enlarge that in just a second. So that light band is called the I band, isotropic. The dark band is called isotropic, anisotropic. So the dark one is the A band, the light band is called the I band. Now I want you to look at the I band for just a minute. You can see the zigzag line going down, and then you see little purple lines going to the right, you see little purple lines going to the left. The same thing is true on the other side. Okay? And then you see the dark band in the middle. Now they're going to take what we have there in that illustration, image number 3, they're going to take it and bring it to a greater magnification. So let's look at the bottom image on page 345, figure 10.6. And now we're looking at the same thing, but we've just got it magnified very much so. So I want you to look over to the right. And you're looking at the eye band. And you see the zigzagging line. Okay. I want you to draw a little number from there. Draw a little line from that zigzagging line. You can put down number 1. And you're going to call it the Z line, or some people prefer to call it the Z disc. Z as in Zorro. Did you guys ever watch Zorro when you were a kid? We grew up with Zorro. Whew. Saving all the good people and getting rid of the bad guys. Anyway, that's called a Z line or a Z disc. Now you notice that as you look at the Z line on the right, you see that little purple line that goes to the left. You can label that number 2. And that's what they call a thin filament. You're going to get some names there. You can put actin, A-C-T-I-N, if you'd like to. That's at one of the myofilaments. So we have myocytes composed of myofibrils, and then myofibrils are composed of myofilaments. Right? So that would be number 2 there would be actin, A-C-T-I-N. 3, you see the little coil coming off the Z line? They come off on both sides. And that coil is another type of protein called titin, T-I-T-I-N. Don't call it T-I-T-A-N. I think Titan was, seems like they were some kind of mythological god or something like that. But it looks like a little spring, doesn't it? But it's another protein. How is that protein different from the actin protein? Go back to chapter 3. Somebody might ask you something about that. And the Z disc also, or the Z line, is another type of protein. Proteins are necessary for life. That's why we've got to eat some chicken and some peanuts and beans and get all the good protein we can so we can make muscles. Excuse me. So we've got 1, the Z line. Number 2 is the actin, that little thin purple line. You see you got 3 of them on one side and 3 of them on the other side, and they cut them short on both ends. So now number 4 is the thick. Kind of looks like could you say it looks like a real used toothbrush, or maybe it looks like a bit of a tree coming out of the ground or something there. Got a bunch of bumps on it, doesn't it? That's another myofilament, and it's called myosin, M-Y-O-S-I-N, myosin. It is a thick filament. It's actin and myosin that actually bring about the contraction, the sliding over of the filaments that we'll talk about in just a few minutes. So those are all protein filaments. And so you have the same thing on the other side. Now, you notice you got a Z line on the left side and you got a Z line on the right side. So draw one of those brackets And from the left Z line to the right Z line is called a sarcomere. A sarcomere. Sarcomere. A sarcomere. That is the contractile unit of a muscle. I was looking this up to see what I could come up with in terms of a number. You and I were looking at a muscle fiber or cell, and we said, okay, we got 10, 12 myofibrils that make up that muscle cell. And then you've got these sarcomeres, which are composed of muscle filaments or myofilaments. And they said a muscle cell can have anywhere from 5,000 to 10,000 sarcomeres. Depends on how long it is. So your muscles in your legs or those muscles in your back that keep you erect when you're walking or you're sitting and you're just standing watching something. Those muscles are having some degree of contraction so that they keep you upright. So depending on the length of the muscle, you can have from maybe 1,000 of them to 10,000 sarcomeres in a muscle fiber. So, or excuse me, I should say in a myofibril. Okay, now let's go over a little bit more to page 344. We're going to look at the same thing, but we're just going to get a little different view of it. So again, you see this in the left-hand side on page 344, you see the same view. And you see it's enlarged like it was over here on the right. And then you look at it down here on the third image, and you see the same thing. And they pull that air out and point toward the myosin filament, muscle filaments. And you see there's a bunch of them in there. They're all put together, got tails. It kind of looks like a little golf club, doesn't it? And then they've got heads. And the heads are going to play a role in contraction. But you see there's a bunch of them there that are intertwined and so you have heads on both sides. You'll see why that comes about, just why that's important in just a few minutes. There's Titan down there. Looks like spring, doesn't it? You guys ever have a slinky when you were a little kid? You would play with that? Yeah, it's a little springy looking piece of protein is what it is. How's it different from actin? How is it different from myosin? And if you look over to the right, you see the purple, looks like a bunch of little grapes there. And the little purple grapes are actin. That's another kind of protein. How are they different? Think about that. We've already been over that. And then you see this blue line on there. And you also see a little yellow group of 3 balls. This is just the way they have illustrated it and put some color to it. So actin, you see it has an active site, got that little depression, and you see tropomyosin twists around the twisting actin, and you see troponin in there also. Troponin, as it is in the particular form in this picture, prevents contraction. But as later, we'll see how it changes, and then we get a contraction. So, tropomyosin covers the active sites of the myosin, and troponin maintains the tropomyosin so you can relax that muscle. Let's look over on page 346. Page 346, and they have another illustration for you. You see the sarcomere from one Z line to another. And you see what they've done is they've enlarged it again. So you get another view. So you see the Z lines on both sides. You see the actin filaments, the thin filaments pointing toward the center of the dark band, or you can say the A band. And then you see the myosin in the middle, got like little heads on the ends. Now notice let's look at the first myosin molecule. And you see that little coil, that coiled protein called titin. Looks like a little spring, and you see it's anchored to the Z line and the myosin. That helps stabilize that myosin protein. I got some other thoughts. I don't know that they've been verified, but that little spring-like structure is kind of like a spring. If you stretch the muscle, it helps the muscle come back to its normal shape. I'm wondering, and I'll mention this maybe later, that if it doesn't have another little function, I could be totally wrong. That's science. You see if you can find out what is right and what's wrong. And sometimes you say, well, I didn't find that. Okay, so it must not be what I thought it was. That's science. That's the way it is. Just because somebody says this is the way it is, 2 years down the road, they'll find out, we didn't know that happened. We didn't know that mercury is going like that. So that's good science. We're always learning how to do things better, and we should. Now, in the middle of the dark band, you see 3 lines running down the middle. Look where the myosin is, and you see behind there 3 dark lines. That's called the M line, as you see. It's in the middle of the H zone. If you look down below, you see underneath that actual electron micrograph of muscle of a myofilament, or a myofibril, I should say. So the M line is another protein that helps to anchor the myosin, maintaining its position. Now, we've talked about what? 1, 2, 3, 4, 5. 5 different proteins that make up a myofibril. Yeah, a lot of protein. We have to have some protein in order to make that muscle. Now, when you and I eat chicken or beef, this is what we're eating. We're eating the myosin, we're eating the titin, we're eating the actin, we're eating the troponin of the chicken, of the cow. You get to go to barbecue up here, Schuler's or Cane's or Schoolhouse Barbecue or something like that. And that's what you're eating. You just happen to be a hog instead of a cow. So you want to be able to identify the structures that compose the sarcomere, those different proteins. Some of them are proteins that stabilize other filaments, other proteins. Some of the proteins are the ones that participate in the contraction that we'll look at in just a few minutes. Okay, so let's go over to 347. And you see the fingers there. Okay. Well, what they're talking about, what they call the sliding filament mechanism. And they used to think that the proteins shorten, but they don't shorten. They don't do that. They stay the same, but they slide over each other. So whenever contraction takes place, the Z lines move in toward the middle because the myosin is pulling the actin filament in toward the M line. Let's look over on page 348. As you look at the top of page 348, figure 10.9. You look at figure A, you see underneath there the little caption says the I band and the H zone narrow during a muscle contraction. Look at the top half of that illustration. That's when it's relaxed. But you see when the Z lines move close to each other, closer to the M line. The actin filaments are drawn toward the M line. The myosin stays stationary, but the actin filaments slide in. So if this was the myosin, here's the actin. Then when the contraction took place, the myosin would move, but the actin would move. So what shortens is the sarcomere. So you could say during contraction, the Z lines of the sarcomeres come closer to each other. However you want to say that, come closer to each other. They come closer to the M line. That's another way of saying it. Because the myosin heads are pulling in the actin filaments. Pull them toward the M line. So the sarcomere shortens, but the filaments themselves do not shorten. It's just the whole structure shortens. Okay? Now, having said that, how do we get a muscle fiber to contract? That's one of their characteristics is contractility. So we look on page 349. We're looking in the first column. We have a little bit of information presented to us a few weeks ago, and we dealt with membrane potentials. Now, remember how we said a student can have a lot of potential? That means there's a lot of possibilities of doing things that are good for the community as opposed to burning buildings down like some of these people are doing. So what we want to do is look at membrane potentials for just a minute. Now, if you come down, you see this little picture, got a membrane, sarcolemma, extracellular fluid, remember that's ECF, and then intracellular fluid, they call it the cytosol of muscle fiber, some people call it intracellular, not intercellular, but the intracellular. Intra means within. So you can call the inside of that, the inside part of that cell is cytosol, cytoplasm, muscle is sarcoplasm. You can call it intracellular fluid. All those terms mean the same thing. Now notice on either side of the sarcolemma, you got positives on the top and negatives on the underside. The top represents the outside, the extracellular fluid, and the sort of pinkish fluid in there is the intracellular fluid, or you can call it the cytosol. And it's a little bit negative right by that cell membrane. Remember, the sarcolemma is the cell membrane of a muscle cell. So as you think about this, the reason there is a positive charge just outside the cell membrane and negative on the inside, that is due to unequal ion distribution. You could also say unequal or unequal distribution of ions or concentration of ions. If we all had the same number on either side, there would be no positive or negative. But since they're not like that, they're not equally dispersed, then you have more positive ions outside than you do on the inside. Therefore, the inside's a little bit negative. At least they represent it that way. So we would say it's polarized. Okay, polarized, like a battery, positive pole, negative pole. We could say it's charged. You guys are going to go to work today. You're going to come home, whatever, and you got to have a battery in that car. That battery's got to be charged, or you're not going to go anywhere. The same thing is true of our muscles. If they're not charged, we don't do anything. You've got to have a charge on those muscle fibers. And that's also true of the nerve fibers too, which we'll work into there in just a second. So the fact that it's charged on both sides, you got a positive charge and negative charge, or polarized, we say it has a potential, an electric potential. You can come down into the third paragraph of that first column, and you see the bold print term. It says electrical potential. It's some potential energy is what it is. When those ions start moving, that's a message that's going to be sent, in this case, to our muscles. And that's why my lips are moving. And you can hear me enunciate words. My tongue's moving around in there. My lips are changing and so forth. My tongue comes against my teeth and so forth. And we make words and we understand what's going on. I'm waving my hands and so forth because that's charged. And there are messages going through my body all the way from here down to here and into your ears because we got charges. We got polarized cells. We have a membrane potential. As I'm talking, my feet and my legs are resting. So if you come now, to the last paragraph, you see the term membrane potential, and you see the term resting membrane potential. So there are no action potentials. There are no electrochemical messages. There are no nerve signals going on in my legs. They're still. They're charged and they're ready to go, though. You and I went to sleep last night. Hope you got to get some sleep. I got up at 3 o'clock this morning. Couldn't go to sleep. So as I lay there, my muscles and my nerves going to my muscles were charged. If the house caught on fire, I'd be bounding out of the bed. And little messages would go through my body, and I would move my muscles and get my wife and grandson and so forth out of the house. Call 911, all of that. The potential to do work, and that would be work in the sense of carrying out some sort of activity. So our cells have to have a resting membrane potential so a message can be transmitted. And you see the word polarized down there at the bottom of that column. Just means you got charges that are different on the outside from on the inside. Let's say 70 out on the outside and 20 on the inside or something like that. It's different. Okay. It's ready to go. What creates the resting membrane potential? It does not happen by chance. Look in the second column on page 349. And I want you to read those first two paragraphs. Now, what I'm interested in you remembering is as you look in the second column there, you see it says the most important active transport pump. Remember how we have passive transport and active transport? Active transport means you got to have expenditure of energy, right? Expenditure of ATP. Look at the first sentence. The most important active transport pump for sodium and potassium is the sodium-potassium ATPase pump. You can just call it the sodium-potassium pump. But ATP is involved in that. So I want you to read that. That protein is a pump. It does not contract. It doesn't transmit a message. Its sole purpose is to maintain the resting membrane potential. In other words, You're loaded and you're ready to go. Okay, now that took ATP right there just to do that. So that pump is extremely valuable to us. It's a protein in the cell membrane, and it transports sodium to the outside and potassium to the inside. And so we end up with a charge. We end up on either side of the membrane. We end up being polarized, positive on the outside, negative on the inside. That protein is very valuable. There are genes in our body that only produce that protein that produces that pump. It's different. The gene is different from the this particular gene that produces a sodium potassium pump protein. Actin, different gene. Myosin, different gene. Titin, different gene. M line, different gene. Z line, different gene. Thousands of different kinds of proteins in our body. They don't just show up by chance. You got to play them. Now I want you to look on page 350. Got about 7 more minutes, and then I'll knock it off, and we'll do another one a little bit later. This is a lot of stuff to take in, but you got to write it down, and you got to start teaching the closet door. Don't wait till next 2 weeks from now and start thinking, I'll study it then. Don't do that. 350. You see the heading says action potential, first column. Key property of all cells, including excitable cells such as muscle fibers, is that the resting membrane potential that's produced by the sodium-potassium pump can be altered to become more negative or more positive. One such change is the action potential. The action potential can be called a nerve impulse. It can be called an electrochemical signal. There's several terms, again, that describe this particular event where the ions move and change. Remember now, the reason we have a polarization, or we have a positive on the outside, negative on the inside, is because we do not have the same number of ions on both sides. If we had the same numbers, we would have 0 for a membrane potential. But because the pump throws sodium out and brings potassium in, we have a polarization. We have a membrane potential, which is normally, they'll say anywhere in this book, they'll go from 70 millivolts to 85 millivolts. So here's a great definition. Look at the components of the definition of an action potential. You see it's quick, extremely quick, like milliseconds. It's temporary. It's a temporary change in a single region of the plasma membrane. Now, it'll spread, but it's got to start in a single area of the plasma membrane. And let's see, there was one other thing I wanted you to know about that. Let's see, it's quick, it's temporary, it occurs in a single region of the plasma membrane. And let's see and I think what I'd like for you to know is that the polarity reverses, the charges reverse, where it becomes positive on the inside and negative on the outside. That little point is your action potential. So you see it says during the action potential, the membrane potential goes from its negative resting value to a more positive value. That means that the inside becomes positive and the outside becomes negative. You see it says this change can be used to signal electrically, to communicate with, and sometimes stimulate a response from other cells. And it says all of this happens in just a few milliseconds. Now think about all the movements you and I do all day long. Think about your heart's got to be beating, right? And you got to be ventilating. Your diaphragm is going up and down, up and down. Your heart is pumping. Last night, while you were asleep. And an action potential has to be generated in order to make the diaphragm move and then relax. Move and then relax. Same thing with the heart. Contract, relax. Contract, relax. Isn't it amazing that the heart contracts and relaxes thousands of times during the day? When you not think of rest, we're thinking 6 to 8 hours, and some of you like 10 to 12 hours in that bed. But split second keeps us going. So I want you to look in the second column, and it's going to mention these words that I want you to know. Second column on 315, you see depolarization in response to a stimulus. That could be you hear a sound and you respond to that sound. This responding means that an action potential has been created and you're moving either away or toward that particular sound. And you could dream up all kinds of scenarios. But in response to a stimulus, don't worry about voltage-gated sodium ion channels. Just look what it says there about sodium ion channels open and sodium ions enter the cell, making the membrane potential less negative. They show it as positive on the inside now. That's called depolarization. The movement of those ions from the outside to the inside is depolarization. And you come down to the number 2 there, you see it says repolarization stage. Sodium ion channels close and other channels for potassium open. Potassium leaves the cell, making the membrane potential more negative. And then the sodium-potassium pump pumps the sodium out, brings the potassium in. That's repolarization. It happens thousands and thousands of times during the day. Now, we're going to stop here on page 352 for this video. I hope I can get another one done made today. But that's on page 352. We'll look at the process of skeletal muscle contraction. Okay? So we're going to end this. And I'll see if I can get another one going while other people are registering some students. I got to register some kids this afternoon. So maybe I can get another one of these done a little bit later. Anyway, there's a lot of stuff, isn't there? Do not wait. Do not wait. You get burned. Okay. So we've sort of got the structure together. Now we'll talk about how the muscle actually contracts, right? All right. Hope you're doing well on your lab exam. I'll catch you later.